Hello, everyone, and welcome to Decoding Disease, How Big Data is Transforming Neurodegenerative Disease Research. I'm Stephanie Ishoo, Director of Marketing and Communications at Target ALS, and I'll be moderating our session today. We'll cover topics like open data sharing, interoperability, AI, and more. We're joined by Amy Easton from Target ALS, Leslie Kirsch from the Michael J. Fox Foundation for Parkinson's Research, and Marc Fiume from DNAstack. Thank you all for joining us. To get us started, I'm going to let each of the speakers give a brief introduction, and Amy, let's start with you.
Sure. Thanks a lot, Stephanie. So excited about today's webinar. So just to start, Target ALS's mission is really to break down the barriers that hold back research. And so one of the things that we're focused on and been focused on for the past 10 years is generating and sharing out big data sets. The reason that we're doing that is because it addresses multiple barriers in research. But the number one is the time it takes to study each gene and each protein. So sharing out big data sets dramatically transforms how we do research. Instead of studying just one gene and one protein at a time in your own lab, now you're sharing out data from thousands of proteins and thousands of genes, enabling you to really accelerate your ability to study all of these things at the same time.
Thanks, Amy. And Leslie, how about you?
Hi, nice to see everybody on this webinar. It's so thrilled to be here with Amy, Mark, and Stephanie. So I'm Dr. Leslie Kirsch. I head up the research data sharing programs for the human subjects data at the Michael J. Fox Foundation for Parkinson's Research. If you've not heard of us before, we're here to cure Parkinson's disease. That's really what we're passionate about. We have been funding large-scale data programs for more than 20 years. In the field of Parkinson's disease, and we are thrilled to have a huge base of users who have come in and taken advantage of the data that we've collected in order to progress Parkinson's disease research. And just thrilled to be here with my fellow panelists to talk all things data with everybody here, since we're all so passionate about the topic.
Thanks, Leslie. And Mark?
Thanks, Stephanie. My name is Marc Fiume, and I'm the CEO and co-founder here at DNAstack. We're a software company that helps organizations like the Michael J. Fox Foundation and Target ALS really amplify the impact of their data by curating and sharing these wonderful data sets with the research community.
Thanks, Mark. And so clearly from those introductions, each of you is involved with curating and managing these huge data sets from people living with rare and neurodegenerative diseases. So let's start pretty simple with just a question for each of you. Can you share why you're investing in big data resources and why is this so important for advancing science? Mark, do you want to get us started on this one?
Yeah, sure. So with our software platform, we help power really it's the back end of sharing initiatives like Target ALS's data engine, which is just curating an incredible collection of really high value data sets to accelerate research in ALS. If you're not already an approved researcher, I'd really encourage you to look at those resources at dataengine.targetals.org. There you'll find some beautifully curated data sets from postmortem tissue analysis, stem cells, and one I'm really excited about is the long-read genome sequencing data set. Amy and her team do a phenomenal job turning around access requests in record time. So if you apply for access to these and other data sets, you generally get access very, very quickly. So why is this so important? You know, like many diseases, there are subtypes of ALS and Parkinson's, and because of resources like this, we now have a generational opportunity to understand those subtypes with molecular precision. And of course, that is the foundation for precision medicine. So we're just so grateful to have partnered with Target ALS and the Michael J. Fox Foundation on these kinds of initiatives.
Thanks, Mark. And Leslie, how about you?
Yeah, Stephanie, it's interesting. I've been at the Fox Foundation for about 5 years now, and in the time that I've been here, I don't think that anybody would say that the research questions have gotten easier. They've just gotten more complex and require more and more data in order to address them. We look at Parkinson's disease, it's the experience of Parkinson's for those that have it, it's extremely heterogeneous. And so with that complexity means that you will always need more and more data, especially in a spot that we are in the field of moving toward biological definition. I either need data sets that are a lot of people narrowly focused on one type of data, or I need to be extremely deep on the amount of data that I have on a smaller set of people to be able to address those questions. And so thinking about it from that perspective, it's just more and more data that we're calling for in order to get to important answers, and also looking at that no single cohort is likely to have all of the answers that we're going to need. So also thinking about big data collection as a necessity from that perspective of making sure that we have the necessary variety in that data at the end of the day.
Thanks, Leslie. And Amy?
Yes, thanks, Stephanie. Really like what Mark and Leslie just said. And just to add on, again, I think Target ALS is really interested in moving with urgency. And so not only moving with urgency, but also helping researchers identify new targets that come from human tissues and human samples, and it really will ultimately translate into new clinical therapies. So this work really helps to accelerate the identification of new biomarkers and therapeutic targets.
Thanks, Amy. And so we've kind of taken this big overarching look at why these resources are important. But I'm going to ask each of you to maybe briefly describe what data resources you specifically have been working on, and also maybe share one of the most exciting findings that has come out of this work so far. Leslie, let's take it to you.
Yeah, we have a lot of different data resources. I think that would be a fair way to describe the data ecosystem that the Fox Foundation and Parkinson's disease is working with. I'll maybe call out three of our largest scale databases, which are some of our most popular ones within the Parkinson's research community. One would be our Global Parkinson's Genetics Program, which is an extremely large database, tens of thousands of genetic records for individuals with and without Parkinson's disease throughout the world. So that is, like I mentioned earlier, we have different types of data sets that we can use for different problems. So GP2 is a great example of, here is a massive scale data set that is an extremely large number of records focused on one specific type of high dimensional data, and that is the genetic data. So that definitely a large and very popular data set within our community. We have one of our newer resources is called the CRN Cloud, the Collaborative Research Network Cloud, which we work with Marc on as well. And the CRN Cloud is a home for deep multi-omics data, on much of it from brain donations, which huge thanks to our community for the resources that they have given to Parkinson's research. But we have a lot of different types of high dimensional data within the omics space that's available from human and non-human records there. And so that's something that we have a lot of specialty researchers very excited who've been coming into that as one of our newer data sets. And I am contractually obligated to talk about sort of the granddaddy of our data sets, which is the Parkinson's Progression Markers Initiative, also known as PPMI. PPMI is an extremely deeply phenotyped cohort of primarily individuals with Parkinson's or at risk for Parkinson's, where we have literally millions of data points available for these individuals, and they have been followed over a long period. Parkinson's, for those that are unfamiliar, is a condition with a long progression period. So there are individuals who have been in the PPMI study for a decade or more. So we have not only extremely deep data on each person, but we also have really deep longitudinal data, which is very difficult to find in this space. And I cannot thank enough our community again for participating. And PPMI has also, for those that don't spend all of their time just looking at news releases about Parkinson's, but you might be familiar with the fact that there is a fairly new biomarker in the Parkinson's disease space around alpha-synuclein, and that was largely driven by those contributions within PPMI. So just study that was set up to find biomarkers, and it might have taken 10 years to get there, but the incredible contributions and the big data that was necessary to do that, we're so thrilled to see our data resources be beneficial to the community like that.
Yeah, that's a really exciting development in Parkinson's disease, and I cannot echo enough your sentiment. We're all, I think, all of us at all of our organizations, just so thankful for the contributions that our communities make, because these discoveries wouldn't happen without them. So with that, Amy, what about you?
Sure. Yeah. Happy to talk about all the data that we're putting into the data engine. These data sets are, again, like Mark said, easily available, and they're supported by the DNAstack's Omics AI platform. We have been generating large multi-omic data sets from postmortem tissue. So all of those data sets sit in the postmortem tissue core collection. You can find literally 5,000 different data points spanning whole genome sequencing to bulk RNA-seq to histopathology that we've collected across at least 500 different cases. So this is a really interesting and unique collection where there's multi-omic data sets that span many different modalities and also really nice clinical annotation of the disease from the people who've donated their tissue. So again, this is what we're talking about when we're talking about big data sets, at least 5,000 data points across these different regions. And so our RNA-seq data collection, our bulk RNA-seq data collection, is really the largest ALS data collection to date, and it's just growing. So I want to point out that this is what we have today, but these data sets continue to grow and grow. And in addition, we have an agri data set. So our ALS Global Research Initiative is broken down into two complementary studies, a natural history study, which collects multimodal data sets over time, and another complementary study, which is focus on outreach to underrepresented communities around the US. And so what we're gathering there is really interesting data around demographics and whole genome sequencing data, and we're investing significantly in long-read sequencing from those data sets. So I'll talk more about that, and Marc will too in just a little bit. But these are really large and growing data sets that will help us understand better, not just genetic contributions, but again, the heterogeneity of the disease, and things like what relates to fast progressors versus slow progressors, for example, in ALS. So again, we have a really rich data set on the data engine. And one of the things I'm really excited about that's coming from these data are, first of all, some of the publications that have come out from Humphrey et al. And more recently Petrescu et al. From the New York Genome Center team and others, showing that you can really start to identify subtypes of ALS by using that bulk RNA-seq data. And so they've already started to describe the different subcategories with respect to things like RNA dysregulation, neuroinflammation, and mitochondrial dysfunction and oxidative stress, which interestingly connects back to Parkinson's disease. Some overlapping biology there. So from those initial publications, you can start to see the window into these different subtypes of ALS, which is really the start to getting to personalized medicine. So more recently, publications are coming out from the spatial transcriptomics data that we've funded, really starting to get more granular and give us a bit more insight into the picture of ALS. So things like changes in vasculature and glia related to vasculature are emerging. Things like renewed focus on oligodendrocytes, which are generating myelin that sheathe our neurons and are so important for neuronal function. So we're seeing new biology emerging, and then we're adding yet another data set from 200 cases that's even more granular, with single cell nuc-seq data from Mount Sinai. So again, with each layer of data, we're getting more granularity into what are the disease mechanisms, and therefore how can we get to personalized medicine through those findings.
Yeah, that's really exciting to hear. I feel like we often, for the layperson like myself, describe a lot of these diseases like puzzles, and we can't, fill the puzzle and figure it out unless we have all those pieces. So really exciting to hear about all that development. And Marc, what about you?
Yeah, I want to first start by congratulating Leslie and Amy. This is not easy work, but as each of them have kind of shared, they're starting to have just an enormous impact, and I think setting a precedent for how collaborative research ought to be done. One of the resources that I'm really excited about is the long read collection, which Amy referred to. One of the major subtypes of ALS is caused by repeat expansions, where you have these sort of genomic stutters. There are stretches of repetitive sequences, and these events, repeats and other complex genetic variants have traditionally been hard to characterize with previous technologies in short read sequencing, but they really come into view with these new long reads. So Target ALS has partnered with PacBio, who is a long read leader in our field, to sequence now more than 6,000 subjects with this new technology. And this is going to allow us to better characterize, what is the pathogenic range of these repeats. Not only that, but they also give us information about structural variation and epigenetic changes that might also impact disease progression. So this is a fairly new data set. There are 194 samples that are already online. We're really excited to see that data set grow.
That's awesome. It's exciting to hear how, as technology is advancing, we're staying like right on the pulse with it to make sure that we're getting the best answers and data and resources for our community and our scientific community to really push forward. And so this world of big data might feel newer to some of us, but it's been evolving and changing over time. And so, Marc, how has data sharing changed among scientists over time?
Dramatically. Yeah, dramatically. So it used to be that scientists would individually generate and analyze the data that they needed for their own experiments. But far too often, those data sets wouldn't be shared. Or worse, their results wouldn't be reproducible. In fact, NIH has found that a large percentage of the studies that they funded, which represent billions of dollars in investment, just aren't reproducible. And so we've seen an awesome shift to a much more collaborative model, which is being pioneered by organizations like Target ALS and MJFF, where the data is purposefully generated as a community resource. That's very different from how it used to be. And there's an explicit mandate for open science and collaboration. So now we have this wonderful multi-institutional international teams of collaborators working all aligned together to generate larger, more diverse, better harmonized, and more easily shared data than we've ever had before. Since the launch of the data engine 2 years ago, we've now registered 500 researchers across very different sectors. So I've never seen anything like the impact that these two organizations are making together. And like I said before, I think this sets a precedent for how we should be doing science.
That's great. And so with all of this connection, the data has to be more connected too. And we've come across this term data interoperability. So Leslie, I would like to ask you, what does that mean?
Well, I guess, Stephanie, maybe I'll start with my highly technical definition that is usually how I think about it in my head, which is data is interoperable if it is easy to smush two pieces of it together. So very, very technical there, I suppose. What that means in practice is that challenges around that usually involve that there are differences in syntax, which could mean that we have the same concept in multiple data sets, but it has different names and labels. A really common example that we see in a lot of data sets around that is something like how sex and gender are represented, right? So it might be the words male and female. It might be called sex in one. It might be called sex at birth in another. It might be zeros and ones that we use to represent it, right? So all of that has to be aligned for data to be interoperable. We have structural differences, so we can have the same data, but it's stored in different ways. Something like we could store it in a long format. So you've got a lot of columns in Excel as an example of a long format, or a Y format, sorry. And you can have a long format, which is that we have a lot of rows, right? So we have a small number of columns and a lot of rows. So simple things like that. Again, just challenges to making it easy to smush it together. And the really tricky thing when we start dealing with a lot of the high dimensional data sets that we're talking about today, we can have semantic differences where data looks the same and it actually isn't at all the same. So we could have different processing pipelines for imaging data, right? Or we could have, in the genetic space, a lot of analysis involves a reference genome. The reference genome isn't the same over time, right? So you can have two sets of data that look like they are exactly the same, but they were processed with different reference genomes. And in fact, they are not comparable in the ways that you might think that they are comparable if you just went and tried to smush them together without first doing your research on how smushable they are. So hopefully, yes, very technical terminology that everybody can leave this session with of smushiness of data.
Yeah, I mean, I think that's helpful, at least in my head, to remember smushing instead of all of the technical terms that would probably go among that. And so recently, Leslie, you published in the Journal of Parkinson's Disease. And can you share some of the challenges with data interoperability and the key takeaways of the publication?
Well, first, I would like to call out one of my amazing co-authors, Amy Easton, for that paper. In that paper, we actually held a workshop, a number of organizations like Michael J. Fox Foundation, Target ALS, who are involved in actively collecting, managing data sets of this type in the neurodegenerative space, or dealing with some of the tools and infrastructure that are related to that. And so we can see that to bring data like this together, set aside the technical smushing things that I just talked about as some of the things that can make it hard. We also have policy and cultural things that can make it a little bit difficult for us to be able to easily bring data together. That we have siloed systems that have to be able to talk to one another. We have what might be slight differences in policy. You know, Marc put it really well that the field has changed enormously around data sharing and the enablement of open data. But a lot of data sets that we're dealing with might, like PPMI, have been collected for more than a decade. And so there might be policy issues that go around that. And culturally speaking, while it has progressed a lot, there's still more progress, I think, to be made about the difference between this is the minimum that it takes to share our data openly, and then it's a much higher bar to make it as effective as we'd like it to be for data interoperability to really be able to talk cross disease and even within a disease. But on the bright side, I feel like there was a lot of enthusiasm and excitement for us to all be working together. We're all seeing how important that is as funders. Our research community is seeing how important it is to be able to do these analyses that cross different neurodegenerative diseases. That's fantastic. I think everybody's also looking at like we don't have to let the perfect be the enemy of the good. We can have pilot programs. We can do things that will make incremental progress, help us learn from it, and just continuously improve in this area. And I think there's also a recognition that there's a lot of different people involved in insights for research conducting with these data sets. And so making sure that we understand how to bring together and enable the different partners that are important, right? So like a neuroscientist is not a statistician. They don't have to be. How can we instead make it easier to bring the knowledge from those groups together and bring it into the data so that you can then use the data more effectively in analysis to move the field forward. So I think I'm thrilled. I just feel, and Amy, I think, you know, you and I have talked about this, that it was just wonderful how much excitement and enthusiasm there is to be working together. It just ain't easy, as Marc, you kindly, you know, called out for us earlier that we are doing an enormous amount of work as organizations and our community is doing a tremendous amount of work, but it's a continuous journey.
Yeah, and collaboration is a deeply held value here at Target ALS. We can move farther faster if we do it together. And so, Amy, what is Target ALS doing to help solve problems of data interoperability? And can you share some of the follow-up from the workshop?
Absolutely. And I guess what I want to call out first of all is what we do prospectively versus what we're doing to address all the big data sets that have been accumulating historically. And so going forward, what I think has been great is that we met Marc Fiume and the DNAstack team just at the right time where they had developed a federated platform to allow for our data sets to be interoperable with many others. I'm not going to elaborate. I'm going to let Marc talk about that. But that was a conscious decision to be able to enable that from the beginning. In addition, when people sign up to access the data in the data engine, they also automatically have access to Verily Workbench at the moment. And Verily Workbench hosts a number of different big data sets, including GP2's data set, all of us, a number of other ones. So we did consciously think about how to make the most of the data that we're generating by allowing it to be interoperable with these other data sets through those existing platforms. But with Leslie's spearheading this effort and bringing a lot of the funding groups together, we have maintained our enthusiasm over the past year, and we've continued to meet to work through a proof of concept example of how we can smush our data together. What do we need to do to be able to do that? And how can we generate an example that others might be able to follow? And in this instance, we're working with folks from Ontario Brain Institute, 10,000 Brains, ADDF, Allen Institute, Michael J. Fox, and Aligning Science with Parkinson's. So it's been a fabulous group of people working steadily in the background to first look at data dictionaries to determine how are we calling different variables? How do we even refer to an individual who's enrolled in the study? Are they a patient? Are they a subject? Are they a participant? Are they just an ID number? So it's unifying across all those different dimensions. How are we referring to ethnicity? How are we calling? How do we refer to their diagnostic category? And allowing us to come to some harmonization around those variables. And then we're going to be working through an analysis across these data sets that have been built, generated, and they're sitting in different platforms. And I think we're really going to be working on different methods of doing this. But one way that's certainly emerging now are AI-based federated learning approaches that allow these data sets to stay where they are. And so AI can go out and analyze data sets in a private and secure way where they are, and then bring the data, aggregated data and results back together in the same place. So this is what we're thinking about, and really excited to continue working on this data interoperability issue, because it's really the foundation for future analyses like AI-based approaches that we really want to enable.
Yeah, and we'll definitely get to AI in a second, but right before we get to that, Marc, I do want to ask you how DNAstack's Omics AI platform helps solve these challenges with the data interoperability, or smushing, as we've come to call it. Oh, I think you're on mute, Marc.
Yeah, so I'm gonna steal that word. Omics AI is a data smusher. It is meant to solve the interoperability problem. So, 10 years ago when we sort of started this, we found that data was inevitably siloed. It was siloed by institution, by state, by country, by device, by cloud, and by indication, as I hope we'll talk about later. And so one option is to ask everyone, hey, let's create a centralized repository of all the data. But as it turns out, this is very large, very sensitive, and very regulated information that we're looking to simultaneously share, but do so in a way that is also responsible, and that creates this conundrum and tension. So we built Omics AI as a sort of interoperability layer for us to be able to create virtual data sets across silos, almost like creating an internet for biomedical research. And this is a big win for data providers who want to share their data, but also for researchers, because while our platform, may host data that lives in different places, it's still, by the magic of interoperability, accessible through a unified interface. So the goal is that on day one, when you get access to these data sets, you can start doing your science without needing to deal with the complexity of interoperability.
Yeah, that's really exciting to see that these solutions are being built, and DNAstack's kind of leading the way there to help us be a more collaborative environment, but keep that integrity of the data and the resources from the different groups that are building them. And so Amy set me up nicely to go into that big question about AI, or artificial intelligence and machine learning. It seems like we can't go a day without hearing about it. But it is, an exciting technology, but also there's potentially maybe some guidelines or policies we should put in place when considering using AI on our data. So Leslie, can you tell us a little bit about those challenges and potential guidelines that we should put into place?
Yeah, Stephanie, I can be the sacrificial lamb on this. So to me, it's very interesting. When I hear people say AI these days, a lot of the time they are talking specifically about generative AI. They are talking about agentic AI applications. But I definitely still think of traditional machine learning and AI approaches as something critical, that I think there's still so much that can still be done that hasn't been settled with those models. But I think the critical thing is that we always have to come back to consent when it comes to the data that we are talking about here, and what our obligations are to those amazing participants who are joining the research. And so it can be tricky with things like generative AI, where using data to train a generative AI model, a lot of the time sort of inherently means that that data is permanently incorporated and redistributed, and that is often not aligned with the promises that were made to participants. So we definitely want to enable these new cutting edge tools, but we do want to make sure that we are thinking about the security, privacy, and consent considerations around it. So I think you see a lot of movement in the space around how do we enable these tools within trusted research environments, right? Where controls are in place so that when the tool is being used, we can be confident that it is meeting all of those requirements. And I think this is definitely something that the whole field is working through as these new tools are emerging, that it's a big conversation in the community. But I think we all come back constantly to what do we owe to our research participants, and what are those consensus we think about how we need to make sure that we are properly applying these tools.
Yeah, definitely. And a follow-up question to that, even with considering that these guidelines are in place, are you seeing AI in the proper ways being applied to your big data resources?
Yeah, and again, I just want to call out, the great traditional machine learning and AI approaches. I think that there are really exciting pieces of research that have been done with going back a few years. So GP2, which I mentioned earlier, that's a data set that was built from the start. The only way you were going to analyze it is with machine learning and AI tools. It's too big to do anything else, really. And there have actually been new genetic risk variants that have been discovered through that program, including one that is in underrepresented research populations, one that is specific to persons of African descent. And that is something that without these large scale tools, there's no way that we would have been able to see that. So just celebrating that there are many, many forms of AI that can be applied to research, and just the great work that's been done for years by those in the field.
Yeah, thank you. And Marc, from your perspective, are you seeing AI being used in the field, and what are you excited about?
Yeah, absolutely. So as Leslie said, that there are real risks to using these models. We all know about hallucination, and I think attribution is going to be an interesting thing for us to work out. But, how do you attribute a scientist that was, assisted by AI and federated learning? Well, I'll park those concerns for now and say overall, I'm very optimistic about our ability to harness this technology for good. The two areas that we're exploring, the first, as Amy alluded to, is federated learning. So we mentioned that we have now this federated infrastructure where we can create these virtual data sets across geographies. How can we now leverage our collective healthcare experiences to drive new insights that were impossible for us to do alone? And I think this kind of machine learning can enable collaboration at planet scale. The second and newer, more gnarly form of AI is in agentic science. How do we think about large language models as sort of a co-pilot for scientific discovery? And so we're doing R&D now on how we can make scientific exploration more productive. For highly trained experts, you could imagine not having to write an SQL statement again, right? That's kind of like long division. It's something that we can do, but we don't like doing, and it's not the best use of our time. So it's those kinds of tasks that we feel like large language models are very, very good at, and we think we can constrain those in the right way that makes them more productive. But that simultaneously makes science more accessible for non-technical experts. Suddenly someone can now express their question in plain English and be able to interrogate the data set without needing to understand SQL at all. And so over time, if we can make science, 3x more productive and 3x more accessible, we can effectively 10x the impact of scientific discovery. And we're so excited about that.
Yeah, that is really exciting. And so we have talked a lot about the technical side, the technological side of this collaboration, but I do want to talk about the human side of data interoperability. And so I'm going to ask each of you how the ability for people and groups to collaborate impact our effort here. And so Leslie, why don't we start with you?
Yeah, so it's always been a fun thing for me in data science that you won't find out what is unclear and challenging successful analysis of your data until other people start using it who aren't you. And so that to me is a huge benefit. And I spend so much of my time, honestly, helping wrench information out of scientists' heads and put it into a place where it can then be fed into analysis models. And it really is uncovered by collaborating around a data set. That is the only way that you learn the quirks of data that impact the ability to draw insight from it.
Thank you. And Amy, what about you?
Yeah, thanks, Stephanie. I mean, absolutely what I've seen over the past 10 years is this really wonderful evolution of people realizing that they benefit, it benefits themselves and it benefits the patients to work together and be more open in their data sharing and being willing to allow their data sets to be analyzed alongside other big data sets. So there are a number of examples of people across the ALS field doing this. And in particular, right now we have AMP-ALS that's gathering really large data sets, some that have not been previously accessible into the knowledge portal. But again, what I want to just remind people is these are existing data sets, so it requires a manual movement of transfer agreements to be signed. And this is true for anyone who's trying to do this right now. So there's a lot of time and resource that it takes to do this manual movement, and also potential for error to be introduced or even privacy gaps when you're moving these data sets around. So again, I really think that everybody recognizes this is not efficient, and the cultural shift is definitely there to work together better to allow for interoperability.
Thanks, Amy. And Mark, why don't you close us out on this question?
Yeah, wonderful. I was actually on a call this morning with one of our other partners, Autism Speaks, and they were excited about a conversation that they're having with Leslie's team, because there's a understudied connection between autism and Parkinson's. So, autism has traditionally been thought of as a neurodevelopmental condition, but it's so nice to see, I call them the Avengers of neuroscience, like the really best minds across multiple indications coming together to understand the brain. And what's amazing is that by virtue of these data being interoperable, we now can collaborate and ask questions that we couldn't before, not just within indications, but across indications. So it's so nice to see good friends come together, and I think we stand to learn a lot from each other.
Yeah, that is really exciting. It is very like small world, right? And so now we're going to move into some audience Q&A. Thank you to everybody who submitted questions ahead of time, and I see a bunch of you are submitting questions now. So thank you for that. It helps shape the conversation. And so first, I think the burning question on a lot of the scientists' minds right now is how can I access these data resources? So we had a little plug for the data engine earlier. Thanks, Mark and Amy. But Amy, why don't you get started there?
Oh, sure. If you just go to Target ALS, data engine dot, oh, what is this actually?
Dataengine.targetals.org.
Exactly, the other way around. If you just go to the data engine website, you can easily get in there. And then what you do is you'll just click on a little black box on the right that will ask you to request access. You'll sign a short data use agreement. And within a few hours to 24 hours, you'll have access to any of the collections that are there. So it's pretty easy to access postmortem tissue core data along with our natural history study data. Just head to that website.
Awesome. And Leslie?
Yeah, so the Michael J. Fox Foundation, and Stephanie, I imagine we can probably hopefully send some links out to the
Yeah,
group afterward.
definitely.
Great. So we actually have a section on our website under our For Researchers section called Data Resources that has links to all of our major data repositories and research gateways. So you can follow the links there to get access, and each of the individual programs has the instructions for how to get access, extremely similar to exactly what Amy just described for your ALS data sets.
Awesome. And Mark, any thoughts on giving access to people?
I will just say, one, it's astounding how quickly you can get access to these tremendous resources. And two, if you're having any trouble at all, either getting access or once you do have access, interrogating the data sets, we're here to help. Please do reach out.
Awesome. And so one other question that's come up a couple times, we kind of touched on it already about with the interoperability and collaboration discussion. Are different agencies, governments, and other groups collaborating to have a larger data pool to work with? I think there are probably a lot of people in our communities who have given data to whatever number of organizations. And so how are all of these groups coming together? Are they coming together? Amy, do you want to start us off with that?
Sure. There's a couple of really great examples. So I just mentioned AMP-ALS. That's an initiative that started a couple of years ago, and it's a public-private partnership with a number of folks involved, industry partners, as well as nonprofits like Target ALS, working together with FNIH and NIH to curate these really wonderful data sets from people living with ALS. And so they're all available in the ALS Knowledge Portal today. And if they're not there yet, they will be. Target ALS's natural history study data are in there. And so NIH has also made the AMP-ALS data sets interoperable, or they will be interoperable soon with the other data, AMP-PD and AMP-AD data sets.
That's great. Leslie, anything to add to that?
Well, Amy already hit up the AMP-PD set, which I was absolutely also going to give as an example. And I think to the question of are there governments and groups? So that's maybe a little bit US centric, but there are other groups internationally who also bring together data sets in different spaces since, you know, different global partners have different regulatory regimes around how they like their data to be shared and where they like it to be stored. But groups like the European Partnership for Neurodegenerative Disease, EPND, you know, that's a place that brings together a large number of data sets on the AD workbench. Not all Alzheimer's disease data sets. They actually have a number of different neurodegenerative disease data sets. So, you know, that's another example again of groups bringing together this data. But I think to some of the conversation earlier, there's a lot of excitement in the space these days as well about learning how we can work together in a federated model rather than having to, as Amy put it, manually move all our data to one place, which can be a much higher bar to clear for a lot of different reasons.
Yeah, and we have our federated data model expert, Marc, here. So anything to add to that, Marc, from your perspective?
Yeah, I just may call out the Global Alliance for Genomics and Health. That is a worldwide organization working to create technical, ethical, security, and regulatory standards for at least genomics and related health data. Those are the standards that we are building our software platform in compliance with. But really importantly, these same standards are being adopted by some of the world's largest precision medicine initiatives in the world, you know, with the aligned view that if we are all compliant with these standards, we will within each other be interoperable. So we're really excited to see this conversation take shape at the global stage.
Awesome. And we also have, as you can imagine, and Leslie kind of pointed to this earlier, not every neuroscientist is going to be a data scientist. And so for people who are entering this field and have like interest in learning more before, you know, we have the AI ability to just ask it a question and it to pull back the answer. Are there any recommendations from you all on how to get started with analyzing these resources? And Marc, if I could go to you first, that would be great.
Yeah, I would just say read the documentation, but don't get stuck. We have you know, teams of data scientists and bioinformaticians who would love to understand your use case and your problem space and to support you. So you're not alone in this mission. We're very happy to help.
Awesome. And Leslie, anything to add there?
Yeah, so I'd point to a few of my favorite resources within the community. So one, if you're not familiar with the organization, the Carpentries, they are an amazing training organization for researchers that can help you get started with software and data tools. So I would definitely encourage, you can check out, they have great self-directed learning if you are looking to get started with a little bit of data science. Again, Marc, like the read the documentation, please read the documentation. We do try to put a lot of effort into our user guides, our getting started workspaces, all of these things. Please engage with them. And if they don't answer the questions that you have as you're getting started with the data set, please, we want to hear from you about them. And at least for those of you that, if you are working in Parkinson's research, we actually have a community of practice at the Michael J. Fox Foundation for those that are working with Parkinson's data, whether it is MJFF funded and sponsored data or not. So you can join our data community of practice and talk to others that are working with data in this space who can help you and collaborate with you.
Awesome. Amy, anything to add there?
Yeah, absolutely. So I would just say for folks who are not hardcore bioinformaticians, but you're curious about what our data sets have to offer, go ahead and get into the data engine, click on the data collection of choice, let's say it's postmortem tissue, and take a look at the metadata tab. So there's a whole genome sequencing metadata tab and an RNA-seq metadata tab. In there, you will find the clinical data. So if you are curious to know what are the diverse ethnicities and races that are captured in our data sets, you can easily see that in the clinical metadata. If you want to know how many C9orf72 repeat expansion carriers are in our data sets, you can easily filter for that. If you want to look for things like comorbidities or family history, there's a lot that you can start to see just in the clinical metadata itself. And that allows you to understand what kind of questions you can ask further, and then you can engage with your bioinformatician friends, either at DNAstack or elsewhere, to dig into the data further.
That's great. Good tip. All right, so we are actually right at time. So thank you everyone for joining us. Thank you, Amy, Leslie, and Marc for sharing all of your expertise in this field. I know a lot of people were curious. And so I will be sending out an email to everyone who registered after the event with all of the great resources we talked about today, accessing all of these great deep data resources, and a recording for anyone who missed it today. But yeah, thank you everyone again for joining us. We really appreciate your time.
Thank you. Thanks, Marc. Thanks, Leslie. Thanks, everyone.
Thank you.
Bye-bye.